Hi everybody, welcome to the second part of the digestive system lecture. So we stopped last time with the stomach. So we have talked about the anatomy of the stomach and what are their functions and also some of the medical conditions of the stomach. So the next part of the digestive system is the small intestine. So we learned the small intestine is broken down to three parts: duodenum, jejunum, and ileum. So the main function of the small intestine is to first carry out the enzymatic digestion, which basically means, again, you're going to require a lot of enzymes, right? Anytime, again, you hear the word enzymatic digestion, you need digestive enzymes. And for that, they work very closely with your pancreas to accomplish that. And then they're also going to absorb all the nutrients that has been broken down into your blood vessels. You also absorb a significant amount of water from your food as the food passes through your GI tract. Now, again, there's three parts of the small intestine. The first part is the duodenum, which is the shortest of the three, followed by jejunum and ileum, ileum being the longest of the three parts of the small intestine. In average, when you look at length of the small intestine, it's about 2 meters when a person is alive, and when they're dead, it's about 6 to 7 meters. So it's a pretty long structure. Now, the reason that it is a long structure is because you basically want to be able to maximize your digestion and your absorption process within the small intestine. Please also do note that you also have the action of peristalsis and segmentation happening, which means you're still going to do the mechanical breakdown through the segmentation process and effectively moving the chyme through your digestive tract using peristalsis movement. Now, again, because we have a small intestine's main function listed as absorption, you want to maximize the amount of absorption you can have. So most effective way to have effective absorption is to increase surface area. So having a really large surface area means you can effectively absorb your nutrients. So if you take a look at your small intestine has about 200 square meters surface area, which is huge considering the size and the folds of the small intestine. So how does your small intestine accomplishes that? So there are two main mechanisms that it does that, villi and microvilli. But on top of that, you also have what we identify as circular folds. So circular folds are these ridges or patterns that you can see in your small intestine, kind of poking out. And these are visible if you take a look at the dissected intestines. The main goal for this is to slow down the food passage through the digestive tract and therefore increase the chance of absorption. So it doesn't really contribute in terms of surface area, but it contributes in terms of, well, it does contribute in terms of surface area, but it also essential in terms of slowing down the food so the chance of absorption increases. Now, if you look at these kind of circular folds that I've depicted, you can see these little finger-like extensions coming off of those, which are kind of zoomed in here. Those are called villi. These are finger-like projections on the mucosa portion, which is basically the innermost lining of the digestive tract, right? These cells are that make your villi are a simple columnar epithelium. But again, think about this. Instead of having, and I'm going to show you, imagine you have this surface area within your small intestine from here to here, right? It's a very small surface area. Now imagine you're creating villi. So what I'm going to do is I'm going to go up and down and up and down. Now, instead of this surface area, what's my surface area? All of these, right? So by moving up, by creating these finger-like extensions, you can increase the small intestine surface area significantly. And again, the more surface area you have, the better your absorption. Now, we mentioned that the villi are made up of simple columnar epithelium, which is kind of showing you right here. So they kind of zoom into this villi. You have these long cells, one single layer, which are your simple simple columnar epithelium. On its surface, you can see these little finger-like extensions. These are on the surface of the cell, and these are called microvilli. And they are present on the apical surface, which is basically the surface facing the food. And again, these finger-like extensions themselves also increase the surface area. So by having these three characteristics of circular fold, villi, and microvilli, I slow down my food and maximize the surface area for the highest absorption rate that I can get. Now, first part of the intestine, as I mentioned, in terms of small intestine, is duodenum. It is relatively short. It's about 10 to 15 inches. It is connected to the liver, gallbladder, and pancreas. The connection to the pancreas is the pancreatic duct that you see right here that empties into the duodenum right here. And you have also a duct called the common bile duct that is coming from both the liver and the gallbladder, and that is also bringing what we identify as bile into our duodenum. So pancreatic duct and the bile duct converge at one point, which is right here, and this point is referred to as the hepatopancreatic ampulla. Again, this is the opening, the enlargement, where the pancreatic duct and the common bile duct enters the duodenum. And we'll talk about why we need to connect pancreas, gallbladder, and liver into our duodenum in just a minute. The duodenum also contains something called Brunner's glands. These release bicarbonate. Bicarbonate is a buffer that helps the duodenum neutralize the acidic environment of the stomach. Again, remember that the only organ that is equipped to handle the acidity is the stomach. So if it enters the duodenum, before that you need to neutralize it. The second part of your small intestine, jejunum, is about 8 feet, and ileum, as I mentioned, which is the longest, is about 12 feet long. The main function associated with them is to, again, continue your chemical digestion using the enzymes as well as absorption of the nutrients. Now let's take a look at the pancreas. So what is the job of your pancreas in terms of digestive system? As we learned, it is one of the main accessory organs we have. It is located right underneath the abdomen, right underneath the stomach on the left upper quadrant. And it has three parts. It has a little tail, it has a body, and it has a head. So a lot of people say it's kind of look like a tadpole. It has both exocrine functions and endocrine functions. What that means is exocrine means functions associated with the digestive system. It is the part that is releasing digestive enzyme into the duodenum for the effective chemical digestion of the food. An endocrine function, if you guys recall, endocrine implies the system that releases hormones. So the endocrine function is releasing two main hormones, which are insulin and glucagon. Now let's focus on exocrine first, which is basically its role in the digestive system. The cells that are responsible for the exocrine functions are about 90% of the pancreas. These cells called acinar cells, and their job is to basically produce, store, and eventually release the digestive enzyme or pancreatic juices into the duodenum. The original enzymes that are produced by the pancreas are identified as inactive and are referred to as the zymogens. However, as they enter the duodenum, they start to become activated and carry out the process of chemical digestion. Your exocrine function also includes secretion of bicarbonate-rich fluid that helps neutralize the acidic environment of the stomach. Now, The function, the endocrine function of the pancreas includes a release of insulin by the beta cells and glucagon by your alpha cells. This should sound really familiar to you guys since we spent a significant amount of time looking at the homeostatic feedback system of glucose earlier in the semester. However, to just kind of introduce that to you guys and make sure everybody's on the same page, let's say we had an increase in our glucose, and again, this is a simplified version of that. If you have an increase in glucose, I should say blood glucose, because that makes more sense. Anything above, typically about 110 milligram per deciliter, your pancreas is going to notice the change. So your pancreas will be acting as both your sensor and your integrator. More specifically, the beta cells of the pancreas are going to notice this, and they're going to release insulin. Insulin is going to be your output, and it's going to send a signal to your effectors. Now, your effectors could be, there are actually multiple structures. It could be your liver. It could be your muscles, typically your skeletal muscles, or basically the cells in your body, as well as fat cells. Oops, fat cells. So the effect or the response as a result of this, when they see the insulin, what they do, they uptake glucose and convert that to glycogen. Convert to glycogen, which is a complex carb, if you guys remember that. If not, please go back and study your organic macromolecule. It's a complex carb. And the final result then would be a decrease in blood glucose. Now, if we're doing the glucagon, I'm not going to rewrite this whole thing. I'm just trying to kind of change some of the labeling for you guys. So if there is a decrease in blood glucose, then pancreas alpha cells are going to release, instead of insulin, they're going to release glucagon, which is going to target the liver, not the muscles. So muscles cannot be a target for it, and fat cells. But what they're going to do is they're going to take glycogen and convert to glucose. Wow, sorry guys. Convert to glucose, and that helps you increase your blood glucose. Wow. So that's the endocrine function. I do expect you guys to be able to recreate this. This is what we learned at the earlier of the semester, so it's just kind of reviewing the same information for you guys. Now, what is your pancreas in terms of enzyme production? What are some of the enzymes? They're not all of the enzymes. In terms of your carbohydrates, which are right here, this category, these are your enzymes associated with carbs. It includes maltose that breaks down, maltase that breaks down maltose into glucose and glucose. Lactase, which breaks down lactose into glucose and galactose. If you guys remember, lactase is an enzyme associated with dairy product. Sorry, lactose is an enzyme associated. Say that again. Lactose is an sugar associated with dairy product. Sucrase is another type of sugar which splits. I'm going to keep saying this wrong, guys. Sorry. Sucrase is an enzyme that breaks down the sugar sucrose into glucose and fructose. And we also have amylase that is responsible for breaking larger carbohydrate to smaller units of carbohydrates or simple sugars. Peptidase is another enzyme that is associated with protein breakdown. You have trypsin and chymotrypsin, which are both associated with breaking down protein to smaller chains of peptides, which are basically made up of multiple amino acids. You have dipeptidase and aminopeptidase that break down and digest your small chains of amino acid to individual amino acids. So logically, again, if you're thinking about this group, their job is to process proteins. And then you have intestinal lipase, which are responsible for breaking down triglycerides into fatty acid and monoglycerides. Again, please do note that this is not all of your enzymes produced by the pancreas, but these are some of the essentials we have listed here. Next, we have your liver. Liver is made up of two main lobes. We have the large lobe, which is the right lobe, and the smaller one, the left lobe. Please do note that the view that I'm showing you here is a posterior view of the liver. This is the anterior view on the bottom. As you can see, this is the right lobe, this is the left lobe, and the line is the falciform ligament. If you turn this around and look at the back, then this becomes your right lobe and this becomes your left lobe. You also have two smaller lobes. You have quadrate and caudate lobe. So quadrate lobe is right here, and caudate lobe is up here, or caudate lobe is up there. You have an extra set of blood vessels, which I'll talk about in just a minute. But here are your hepatic veins, right here and right here. These are your two main hepatic veins for right and left, which empty into these large blood vessels at the center, called the inferior vena cava. FYI, if you guys hear the word hepatic, hepatic implies that is associated with your liver. If you take a look at the microscopic anatomy of the liver, you see this hexagonal structure called the lobules, and they're the ones that basically carry the functions of your liver, which includes about 500 functions within your body. Here's another zoom-in picture of the liver. Again, this is the posterior view. So we have the right lobe, we have the caudate lobe on top, quadrate lobe on the bottom, left lobe right here, and then falciform ligament that secures your liver to the diaphragm. You have two main blood vessels, hepatic veins, that empty into this large vessel called the inferior vena cava. You have another set that are depicted in red. These are your hepatic arteries. Again, you have right and left. You have, in terms of your liver, you have a third blood vessels right here called the hepatic portal vein. The job of hepatic portal vein is to collect a nutrient-rich blood from your stomach and your intestine, bring it to your liver, as you can see, and basically complete the detoxification of those nutrients before they enter your main bloodstream. So one quick note on that, guys. Understand that not everything your body is absorbed is something that can be good for you. You can absorb toxins, unwanted material into your system. So you don't want those to be circulating throughout your body without basically being checked. So what is the job of hepatic portal vein? It goes to the stomach and intestine, collects the blood that was that received nutrients from your digestive tract, bring it into your liver for the purpose of detoxification. Again, we also talked about what you see in red here. These are the hepatic artery, which are responsible for basically bringing oxygenated or O2 rich blood to your liver. And then the right and left hepatic veins, which are right here and right here. They're the ones that take the deoxygenated blood or oxygen poor blood away from your liver. Right at the liver, you also have this kind of centralized space where all of the blood vessels and nerves enter or exit your liver, called the porta hepatis, which is again depicted right here. So that's your porta hepatis. Now, going back to what I was mentioning earlier, so if you're looking at your liver microscopic structure, we have these lobules, these hexagonal structures. This hexagonal structure looks like this. Again, you can see a very advanced network of blood vessels through these lobules. The red ones are still gonna depict associated with hepatic arteries. You have the blue ones that can also, they can be associated with hepatic portal vein as the one that are responsible for detoxification. And you can have hepatic vein, which are basically picking up the blood after it has been detoxed and drop them into the inferior vena cava. You also have these green ducts inside the liver called the bile duct. They're the one responsible for picking up the bile and transferring it eventually out of the liver, combining it with the duct of the gallbladder and emptying it into the duodenum. So what are the functions of the liver? So let's take a look at them. Liver is responsible for production of bile, which is responsible for emulsification of fat. It stores vitamins. These are fat-soluble vitamins, which are D, A, K, and E only. So B and C are not included as they are water-soluble. You have processing of fats and amino acid, which is going to be hinted at later in metabolism lecture. It detoxifies any unwanted material out of the blood. And it also produces blood proteins called albumin, globulin, and fibrinogen. What are the jobs of these? Albumin helps you maintain your osmotic pressure for the blood. Globulin is a transporter. And fibrinogen is essential for blood clotting. So even though these are proteins found inside your bloodstream, your liver is responsible for production of them. Looking at some of the liver disease, we have cirrhosis of the liver, which is basically the cells of the liver called hepatocytes are damaged. And when they are trying to be replaced by a scar tissue, they get replaced by fibrous tissue, which is basically collagen. Now, this changes in the actual anatomy of the microscopic anatomy of the liver can cause compression of the blood vessels, such as the blood vessels associated with the portal vein, causing what we identify as hepatic portal hypertension. Hypertension simply means high blood pressure. It can compress the bile duct, which are responsible for production of the bile, therefore limiting how much your body can produce bile and therefore digest and emulsify fat. The main reason somebody will see cirrhosis is because they are either identified as alcoholics or they have some sort of liver disease like hepatitis. The symptoms of cirrhosis is typically jaundice, which is the yellowing of the skin, and the white of the eye, which is called the sclera, because we have a build-up of bilirubin in our system. If you don't know what bilirubin is a byproduct of destroying red blood cells, and the color of the bilirubin is yellow. So if you take a red blood cell, one of the main components inside of it is heme, and when heme gets destroyed, it becomes bilirubin, which is color of it is yellow. And the job of your liver is to basically, it goes to your liver, it goes through your intestine, and eventually basically removed as feces. Now, if your liver is not working fine, bilirubin stays in your system, which causes, again, because it's yellow, it's going to cause the yellowing of the skin and the sclera of the eye. You can also have edema as a sign of liver cirrhosis. Remember that what we mentioned here, one of the proteins your blood makes is albumin, that is produced by your liver. So basically, if your liver is not producing enough albumin, so there is a decrease in the albumin level, then the osmotic pressure will decrease. So there is a decrease in albumin. Basically, I'm going to write it down for you guys. Albumin, which causes a decrease in osmotic pressure. That causes the water to move from blood into the tissue. And kind of think about it. If you have a lot of water in your tissue, it causes what? Tissue swelling, which is also known as edema. Please make sure these two concepts are clear, as I do want you guys to understand why somebody who has cirrhosis of liver have symptoms of jaundice and edema. Hepatitis is a result of a viral infection caused by a various strain of the hepatitis virus. There are typically 6 versions or strains of it, A through F. The ones that you may have heard of it is A and E, which are as a result of contaminated food. It's highly contagious, however, it's also treatable. You also have hepatitis B and C, which are transmitted via blood. Hepatitis B has vaccination against, however, they're still working toward hepatitis C vaccination. Please also do note that there is vaccination for also hepatitis A and E. Gallbladder that is tucked in underneath your liver is responsible for releasing the stored bile. It does not produce its own bile, which is a really, really important thing I want you guys to understand. Liver is the only organ that produces the bile. Gallbladder job is only to store and concentrate the bile the liver has passed on to it. If you have too much bile in your system, which is basically produced from cholesterol, these bile salts can crystallize and form what we identify as gallstones. What they can do is they can block the little duct you see right here, this duct right here called the cystic duct, which passes the bile from the gallbladder into the duodenum. So you kind of imagine you have a little stone down here, right here, and things are trying to get out of the gallbladder. That's going to be painful. So typically, especially if you have fatty food and you have gallstones, you would feel severe pain in the upper right quadrant of the stomach, upper right quadrant of the abdominal cavity as the pressure is placed on the cystic duct. Typical treatment for a person with a gallstone is to have them do a surgery, which basically removes the gallbladder completely. People do live just fine without a gallbladder, but they might have they might have to limit their consumption of fatty food because now they don't have that store of extra bile that is stored inside the gallbladder. Now, one last thing before I kind of switch gears to the large intestine. I keep talking about bile and the word emulsification, but what does that exactly mean? So understand that 60 to 70% of your body is water. And fat and water do not go together. So if you have a fatty food or you have consumed fat, when they introduce into the digestive system, they try to avoid the water environment, so they join each other. So if you have fat, the fat will appear as more of a large droplets of fat rather than little tiny structures, because they all basically join each other to try to limit their interaction with the water environment of the intestines. So when you have a fat or lipid that is about this size, let's say, it has this much of a surface area. So not much surface area. Now, if you limit surface area, so decrease in surface area means that the enzyme has lower chance of digesting this fat, right? So how can I fix this problem? So lipid can be mixed with bile, right? That comes from your liver and or gallbladder. Now, what happens is now that lipids can be broken down to little droplets of fat, and each of them are going to have their own surface area. This means increase in surface area means what? An increase in your digestion. So it is really important to have that bile, because without that, you will not be able to effectively digest your fat and therefore receive the nutrients that are associated with fat. Now, last part of our digestive system, we have our large intestine. A few points about large intestine. It is essential in terms of absorption of water and some electrolytes. You have significant peristalsis movement, because basically you want to collect everything and push them out of the body. You do have what we identify as a bacterial flora that it helps with digestion of some of the indigestible material. And basically the goal is for your body to remove any indigestible chyme from your system. It starts with the cecum, which is this pouch you see right here. Oops, I don't know what happened here. So it's this little pouch right here, that's the cecum. It's connected to the ileum, which is right here. So ileum would be right here. Then food moves up through the ascending colon, goes across through the transverse colon, goes down through the descending colon, twists through the sigmoid colon, and eventually through the rectum and anal canal and anus. The anal sphincters, or there are two sphincters. You have the internal one, which is made up of a smooth muscle. This is the one that basically keeps the fecal matter inside. The movement of this is involuntary. And as the picture is depicting, is the external sphincter, which is a skeletal muscle, which basically decides when you are pooping, for a better lack of like a simple way of saying it. Now, at the beginning of the cecum, you have this kind of extension, finger-like extension or a little worm. That's the falciform appendix. Simply, people call them an appendix. It is part of the lymphatic tissue that's found within the digestive system, responsible for destroying pathogens that may be introduced into the digestive tract. Some of the features of the large intestine, as you can see in the picture, so I'm just going forward, is this kind of pouch-like thing right here, kind of like those. These are referred to as the hostra. These pouches are created by these smooth muscle kind of line or strip you can see going through, this white strip going through, called tinea coli. So again, kind of going back to what we're talking about, tinea coli is the strip of light, smooth muscle that causes these puckering, and these puckering are basically your hostra. This is helping basically again the food to effectively pass through the large intestine, absorb anything that you need, and eventually remove the unwanted kind or unwanted material through large peristaltic movement. Okay, I believe I'm not going to go over this slide. We will talk about the digestion and absorption a little bit more in terms of metabolism later on, but that's about it. The only part that I do want to talk about before we finish the lecture is this part, which is lactose intolerance. This is something that we have discussed previously, but kind of bringing it up again. If people lack the enzyme lactase, they will not be able to break down the sugar associated with the lactose coming from dairy product. The typical symptoms of these individuals is gas production, bloating, flatulence, and cramping. Again, the lack of ability to digest that means the sugar lactose stays inside their system, which basically pools water. It gives the bacteria in the intestines something to break, which is the sugar, which causes a byproduct of gas formation, which causes also the bloating and flatulence. And because you also have that sugar in your system, water gets pulled inside the digestive tract, and that causes the symptoms of diarrhea. I think that is about it. So guys, make sure you guys are continue working on your homework, and let me know if you guys have any question about your lecture.